Thanks for tuning in to BEaTS Research Radio, a podcast and YouTube channel that aims to disseminate science and research to the community. My name is Nicole Chu, an undergraduate student at the University of Ottawa, and I'll be your host on today's episode. Joining us is Dr. Michael Hildebrand, an associate professor at Carleton University in the Department of Neuroscience, an affiliate investigator at the Ottawa Hospital Research Institute. Founded in 2013, Dr. Hildebrand's translational research program is addressing the urgent need for safe and effective new treatments for chronic pain. Dr. Hildebrand's research on spinal mechanisms of pain combines rodent and human spinal pain models to characterize the regulation of voltage-gated channels and excitatory glutamate receptors in dorsal horn neurons and how they contribute to chronic pain. Thank you so much for joining us today.
Thanks, Nicole. It's a pleasure to be on BEaTS Research Radio today with you.
We're so happy to have you on our channel. So to begin our conversation, can you please give us a brief introduction on the research areas which your lab explores?
Excellent, sure can. So we kind of have two main branches in the lab. The first branch is really looking at, as you were saying, these spinal mechanisms of pain and how they work at baseline. So basically how they're involved in good pain. Because even though we think of pain as a bad thing, it's actually really important and protective to help us avoid future injury and to protect parts of our body that are injured. And so when we're investigating these mechanisms, my lab is really looking kind of at a molecular and cellular level to really characterize certain types of proteins called receptors or channels, and how those proteins are involved in regulating the excitability and the signaling between neurons within these spinal pain signaling networks. And as we're trying to understand that, we're really looking at how these mechanisms are potentially the same or are different across critical variables that we know can affect potential translation into new treatments. So things such as how they change across development or how they change across sex for male or females. And then really exciting as well, how they change across species. So a lot of fundamental research is done in rodent models. Through a collaboration with Dr. Yves Tsai at the Ottawa Hospital Research Institute, we have access to human tissue from human organ donors. So we can test whether some of these mechanisms and these different channels, if they're the same from rodents to humans. So that's the first, sorry, I know this is a bit long-winded, but that's the first one, is how pain works kind of at baseline, good pain in the spinal cord. And then the second main branch of our research is how do these signaling mechanisms and this regulation of pain processing at the level of the spinal cord, how does that change to drive chronic pain, to drive basically out of control signaling in the spinal cord that leads to chronic pain? So those are kind of two of the main branches.
Wow, thank you so much for sharing these two main branches. So you mentioned the big overarching focus of your lab is actually chronic pain and a good pain in the spinal cord. But why is chronic pain such a big public health crisis in Canada currently?
Yeah, that's a really good question. I think one of the main reasons is it's so prevalent. 1 in 4 to 1 in 5, and actually those numbers are outdated in the United States now. They have recent studies saying as many as 1 in 3 people suffer from chronic pain. So it's highly prevalent. I'm sure you in your life, me in my life, we probably have either friends, family members, or ourselves that suffer from chronic pain. So I think it either directly or indirectly impacts us all. And so it's highly prevalent, it's highly debilitating. Obviously, it costs from an economic level, it costs the Canadian economy billions of dollars in direct healthcare costs, as well as lost worker productivity costs. But more, I guess, important is just that personal impact it has on those that suffer from chronic pain. And part of the reason why it can be so devastating is because we just really have a lack of safe and effective therapeutics for pain. And I think an example of that is the current opioid crisis that we're in that the treatments that we do have that work really well for acute pain, for kind of short-term pain, such as the opioids, they don't work well for chronic pain. And so that can lead to issues with those that have to take it, and as well as other societal issues that link to opioids. So part of my research then is the kind of big picture goal that we're kind of a part of is trying to figure out what's wrong so we can identify new targets and potential new treatments for chronic pain.
And in this high prevalence, this chronic pain public health challenge, why are women disproportionately affected compared to men in chronic pain cases?
So I think part of it is many of these conditions, many chronic pain conditions are more prevalent in women than men. So examples like fibromyalgia, migraine, irritable bowel syndrome, and many other conditions are more prevalent in women than men. And so the typical chronic pain patient is a woman in those conditions. But actually, why? We don't really, so that kind of shows we don't really fully know the underlying reasons. It could be a combination of factors, like there could be differential environmental factors, societal factors that differentially impact sexes, but there also could be underlying biological differences that are occurring in women versus men. And so that's why I think it's so important is research. A lot of the kind of fundamental biomedical research we have to understand these kind of conditions like pain are based in bias towards males. And so our kind of fundamental understanding is based on the male biology. And so we have a lot of catching up to do to figure out what of our understanding is the same from males to females, but what's different. And so that's part of the type of research that my lab is participating in.
Oh, and maybe like diving into your research, one of your recent publications in the journal Brain focus on investigating pain signaling methods in the spinal cord. Do you mind explaining like maybe the light bulb or eureka moment when your team recognized like that mechanisms of spinal
Yeah.
pain differed between sexes?
So like a lot of science, we kind of stumbled into it. I can't claim any grand design and strategic hypothesis up front. Actually, we were guilty of doing what many in the field were doing. We were starting in male rodents, and partly just because you have to start somewhere, we were trying to base our kind of discoveries on what had been done before. And to keep it simple, we were starting at least in males. We wanted to move on to females as well, but we were starting in males. And so we had identified this mechanism in the spinal cord that was linked to chronic pain in our male rodents. And so we wanted to investigate whether the same mechanism was occurring within the human tissue that we had from organ donors, which was really unique. And so as we were doing that, we saw, yes, there's this effect. Certain proteins were going up, certain proteins were going down, similar to what we had saw in the male rodents. And so we were getting really excited. But then we increased our number of samples. So we did the repeated experiments with tissue from new donors, and the effect sizes started to go down. And so I can remember, because it's one of those moments, you have certain moments in life where it really stick, because you know they have such profound impact on kind of your future. And this was one of those where I was driving home and I was thinking, well, why would that be the case? We have more and we have this clear effect and now it's going down. And then it was like a duh moment where it's like with our organ donors, we had both males and females, men and women. And so I ran to the data, separated it by men and women, and saw this clear effect, this clear mechanism that was occurring within the samples from the men, but not in the samples from the women. And so that was the starting point for many experiments. And I say but a lot of this was done directly in partnership with students in the lab, and the main student being Anne-Marie Dedek, which is a PhD at the time student in the lab. She's now a postdoctoral fellow, but she was the one then that we used this initial discovery of this difference to then do a deep dive into where these mechanisms different in males versus females in our rodent models. We had to go back to our rodent models and more experiments in our human tissue.
Oh, wow. And you mentioned before earlier in our
Yeah.
conversation how critical like the access to these human spinal cord tissues, samples from like organ donors were towards your study. And do you mind like expanding on that a bit more?
Yeah, because it's critical for that translational divide of we have all this understanding in rodents, we want new treatments for humans. And so there's an assumption that those mechanisms will be conserved, but that assumption remains untested most of the time. So most drugs fail in clinical development. And one reason why could be because maybe there's differences in the underlying biology between our preclinical rodent models and our human models. And so because we have this tissue, we're able to apply the same approaches that we've done in our rodents, like we do staining for where the different receptors are, how much of the receptors are. We can do electrical recordings to look at the activity of those receptors and how they change in chronic pain states. And so the same type of techniques that we were doing in our rodent models, we can do in our human models. And that led to some of the discoveries of the paper.
Oh, speaking of the discoveries, for those who may not know, do you mind giving like a general overview of the major findings from their
Yes.
study?
Yeah, no, that sounds great. So the main, to just kind of try to keep it simple, the main finding is we identified, at least in males, we have this pathological mechanism where in these spinal cord pain signaling neurons, you kind of lose the brakes on excitability, and that's referred to as inhibition or synaptic inhibition. So you lose synaptic inhibition, which is the brakes, which would increase excitability. But more than that, the loss of the brakes actually directly causes an increase in the gas pedal mechanisms, increased excitability, which are those excitatory glutamate receptors. And so it's this vicious cycle where you have less brakes, more excitation, and it actually feeds back. It's worse than that, where the increased excitation causes a mechanism that directly feeds in. And so you have this loop where the neurons start firing and sending pain signals more and more and more, which then go from the spinal cord up to the brain, which lead to that chronic pain. But that's in males, in male rodents and human tissue. What this paper showed is that if we did the same kind of approaches, the same studies, experiments in females, this whole mechanism is not there in females in both our rodent models and our human tissue models. So it looks at least for this mechanism at the level of the spinal cord, there's a major sex difference in what's driving chronic pain in males versus females. And then the final experiments in the paper showed that that difference is mediated by female sex hormones. If you remove the sex hormones in females in our preclinical, in our rodent models, it actually caused the spinal cord neurons, once those rats matured into adulthood, to look more like male neurons. They had this mechanism, even though they were from female rats, they had the male-like pathological mechanism once you remove the effects of sex hormones, at least in late development.
Well, that is so interesting to see that feedback loop in the male spinal cord pain processing.
Yeah.
So why is it just so important to investigate these neurobiological differences of pain processing between sexes?
Yeah, I think it really helps us understand what is the same. Because a lot of it, like this is one example where there's major differences. Obviously, there's a lot of elements and a lot of signaling related to pain that's the same. So it's critical to identify what's the same, but then what's different. And that knowledge can then be used to help even inform our current treatments. Because if, for example, if there's a drug that's acting on one of these signaling molecules that's different in males versus females, well, maybe that drug will work better in males or work better in females. So it can inform our current therapeutics in terms of how we prescribe and administer. And even these results have implications for that. It can also, as we see these differences in our preclinical models, it can help encourage and drive the inclusion of sex as a biological variable in clinical trials. Because actually in clinical trials, even, and only in the last few decades have males and females been included on all clinical trials. But even when that's the case, it's often not separated out as a variable. So even a lot of the drugs are failing in clinical trials, but it might be that for some of those, they were actually working in one sex or the other. So as we have this preclinical evidence, that might help drive to have consideration of sex as biological variable in the clinical trials, which could then hopefully lead to better treatments.
Oh, so you've mentioned like the clinical trials and the current possible drug prescriptions, but what are, for the final question, like the future implications of this research for better drug development?
Yeah, and I think one of the hopes of this is that it'll lead to the identification of new targets. So new therapeutic targets that can help dampen this pain signaling, whether that's at the level of the spinal cord as I study, or at the level of the brain or in the periphery, but new targets that will maybe work for both sexes if it's a target that's conserved, or as well, potentially sex-specific drugs that can work for certain syndromes that maybe are preferentially found in one sex or the other. So it's really, I think, the starting point and hopefully the building blocks to help lead us closer to new treatments that work and help alleviate chronic pain for both men and women.
Oh, thank you so much, Dr. Hildebrand. We'll definitely keep an eye out for more translational research coming from your lab.
Thank you so much. It was a pleasure. And thank you for inviting me to be a part of this.
And on behalf of our entire BEaTS radio team and our radio director, Dr. Emilio Alarcon, we thank everyone for tuning in. And we hope to see everyone all again next Wednesday.